right. So just as a reminder where we left off on Monday, we began talking about fatty acids, and we looked how we can systematically name any fatty acid tail or any fatty acid really in general. And we looked at what happens when we change our fatty acid tails in terms of melting point. How does melting point change if we have double bonds? How does melting point change if we change the length of the tail? So on and so forth. And we left off talking about our triacylglycerols, how their energy storage. And we talked about our lipocytes, which are cells that hold our tags or triacylglycerols. And we talked about how triacylglycerols are our most highly efficient energy storage because they don't have to retain water. They're nonpolar. And the bonds themselves have a lot more energy than a glucose molecule, for example. Right? So just to start us off with is our first question of the day. So usually we use triacylglycerols for storage. You don't usually find triacylglycerols in the lipid bilayer. Based on your knowledge of the lipid bilayer, why would we not find triacylglycerols there? Anyone have any ideas on that question? Why don't we find triacylglycerols in the lipid bilayer? Yep, so glycerol, the glycerol head group, that's not really a polar head group. We mentioned on Monday that you do have the C double bond O, and that is polar somewhat, but not really all that polar. Especially at the top of your head groups, you have these methyl groups. So the glycerol head group is not polar, and to be in the lipid bilayer, you really need a polar head group. So that is why we do not find tags there. That's why they're only really used for storage. So let's actually talk about some of the lipids, other types of lipids that we have in our body. So what we're looking at first are glycerol phospholipids. What a glycerol phospholipid is that you take that glycerol head group that we talked about and you add a phosphate to it. So here's glycerol 3-phosphate, and the lipid would add right onto that phosphate. Or sorry, my bad. The lipid is lipid tails right here. This X is showing different modifications that can be added to your glycerol phospholipid. So these are modifications. And based on the modification, you get different names of that group. So you can just add an H there. So that's more or less just a water group. And you have phosphatidic acid. Phosphatidylethanolamine, you add ethylamine group. And the naming system more or less follows that pattern. It's phosphatidyl. Then it just says, what are you attaching to your phosphate? What type of head group are you attaching to your phosphate? And here it's just a list of different head groups that you might find: ethylamine, choline, serine, myo, inositol, glycerol, phosphatidylglycerol. So that's just one type of lipid there, where we have a phosphate, something's usually attached to that phosphate, and then at position 1 and position 2 of the glycerol, you'll just have fatty acid tails like normal. So that is what a glycerol phospholipid is. Now, just as a general biochemistry question, since hydrogen bonds is probably the topic people struggle with the most, I do have a question here. Which head groups can form hydrogen bonds? And I think I have a poll for this. Yeah. Yeah. So here's a way to not only force you to look at these groups, but also testing your general biochemistry. Which head groups, and so it's going to be phosphate blah, and I want to know which of these head groups are going to be able to form hydrogen bonds. So select all the ones that can form hydrogen bonds from these head groups. While you look at that, I will answer B. So various autoimmune diseases that recognize DNA can also recognize phospholipids. What must they be binding to? So just as a reminder, right, DNA has phosphate backbone. So every DNA has a phosphate in it. Therefore, for B, if you're recognizing DNA and a phospholipid, you are binding to the phosphate. So that's the answer for B. And I'll give maybe a minute more for the question that I have up on the poll. Can I draw the last one? It's cut off. Oh, yeah. So on the bottom there, the only thing that's cut off is it's a glycerol. So like coming off of that carbon is another CH2O C double bond OR. So yeah, nothing. It's more or less like a you have a second glycerol coming off of that phosphate. Can I go over B again? Sure. DNA has phosphate in its backbone. So if I'm a antibody that can recognize a phospholipid in DNA, the thing I must be binding to is phosphate. All right. Interesting. So the actual answer is everything except choline. All right. So water. So I'll show you the logic for this, right? So if it's P double bond O OH, I can hydrogen bond to that OH. Ethylamine has that NH3 plus. I can hydrogen bond to that NH3 plus. Choline, the charge group is this, right? Then you have some carbons. This nitrogen is charged, but it is bonded to 4 carbons. Therefore, it has no place left it can actually hydrogen bond. Carbon and hydrogen cannot hydrogen bond itself. So that can't hydrogen bond. Serine has COO minus. Oxygen can hydrogen bond. Myoinositol, I'm surprised by the number of people who do not think that can hydrogen bond. You have nothing but OHs on this, right? Oxygen, hydrogen connected to an oxygen will always hydrogen bond. So that has hydrogen bond groups. Same with glycerol right there. There's hydrogen bond. And phosphatidylglycerol has that negative oxygen right there, which could be in a hydrogen bond. So there's our little reminder of what can and cannot form a hydrogen bond. Remember, it's a hydrogen that's connected to like a nitrogen, oxygen, fluorine, but you're not really going to get a hydrogen there. Or it's a group that's negatively charged like oxygen. So that was just a way to force you to look at these pictures. Any questions about our head groups? Any questions about glycerophospholipids? And I'll clear all that. So everything but choline. While we're looking on this slide, you should know these groups. You should be able to tell by looking at the head group, was a phosphatidylethynolamine versus a phosphatidic acid versus a phosphatidylcholine. It's good to know these different types of head groups. Hence the reason why I wanted you to look at these for an extra couple minutes. As always, I can't force you to draw anything though. I can only ask you to recognize if given a picture. All right, moving on then. The next type of lipid is a sphingosine. A sphingosine is the major membrane component. So in your plasma membranes, the majority of lipids in there are the sphingosines. And what a sphingosine is, take a look at this weird head group. You have CH2OH here. Then you have your nitrogen, and here's your fatty acid. So the fatty acid is on carbon 2, right? Carbon 2, you have your fatty acid that's connected to a nitrogen. Then at carbon 3, you have this constant region where you have carbon that's double bonded to another carbon. You have 12 CH2 groups and 1 CH3. So this carbon, you can either call it carbon 3 or carbon 1, to be honest, but I'm going to go left to right and go 1, 2, and 3. Carbon 2, that's where you're going to have the variety of what type of fatty acid. How many, like, is it C12, C14, C16? That can change. The fatty acid tail on carbon 3 is always the same. That's why it's actually drawn out for us. And at carbon 1, we just have an OH. So this is a major component, the sphingosine. And that's the structure of the sphingosine. And up above here, if you're wondering, that is just like the head group without the fatty acid tail. So this whole thing is the head of the sphingosine. And the most common sphingolipid is the sphingomyelin or sphingophospholipid. So a sphingomyelin is kind of like a combination of the glycerophospholipids and the sphingolipids. Not quite. It's not an exact one-to-one comparison, but we do have the return of our phosphate. So in our previous slide, we saw that this was an OH, right, at carbon 1. Here, we're replacing that OH and putting a big old phosphorcholine group where we have the phosphate and choline. So again, that's why I want us to know what these head groups look like so we can easily point them out. At our, in the text is probably being cut off for you, but this says this is a sphingomyelin. At the C2 position, we have palmitate. So again, it's a constant lipid at C2. And at C3, we have the same thing as we just saw on the previous slide, right? So everything in black is like the head group. So black is head group. The red is our fatty acid tail. And the blue is more or less what makes this a sphingomyelin. So blue, the SM means sphingomyelin. I just didn't want to write it out again. So the blue is like the specific head group for the sphingomyelin there. And as it says on the slide, this roughly makes up 10 to 20% of the plasma membrane. Now, we can continue to switch the head groups at position 1, and we make different types of lipids with that. So we can make a glycosphingolipid, right? Let's break down that word, and if you know, to make it easier to remember. Glyco means sugar, right? So sugar. The sphingolipid is what we've been looking at the 2 slides previously, which is more or less in black on this slide. Another word for a glycosphingolipid is a cerebroside. And this is just take your phosphatidylcholine and just put a sugar on that. And we can even get more complicated sugars, and we call that a ganglioside, where you have this massive sugar attached. So I know the picture's a little cut off, but again, black here is our normal sphingolipid. And the green is our salic acid. So here's a salic acid. Whoops, I don't know how to spell ACID. That's C16. It's a little hard to see. And then it goes CH3 on the bottom there. And then this whole blue and red, that's your sugar head group. You can see compared to the glycosphingolipid, which just has 1 sugar, the gangliosides have a lot of sugars. These will be on your cell surface, and 6% of all lipids in our brain are these complex gangliosides. So again, what I would ask for you when it comes to like a test time, like what information should I know, right? You should be able to know what's the difference between a sphingomyelin, a cerebroside, and a ganglioside. And generally, which one's more common? The sphingomyelin is the most common in the plasma membrane. Ganglioside is somewhat common in the brain. Stuff like that, like just the information on this slide. Any questions about any of the material we covered or what I might be looking from you, anything like that? Flashcards might be key for something like that. This is just brute memorization, which is most of biochemistry. What compounds? Basically every single picture we've gone over today. What does a sphingomyelin? Let me get rid of this. What does a sphingomyelin look by itself? Whoops. Sorry. Let me actually get the question. Sphingomyelin's on the left. This is a sphingomyelin. This whole thing is a sphingomyelin. Are cerebrosides also found in the brain? I think so. Let me just double check that really quick. Yeah, so cerebrosides, not on here, but it's looks like they're found in nerve cells. And also muscle cells is where you're going to find these in the membrane. So they'll coat the nerve, so important for your brain and your nervous system, and also in your muscles. And in the brain. Yeah, so they're very abundant in the brain too. The white matter of the brain in your myelin sheets. Or are the photos on the right? Though the photo on the right here, this is a ganglioside. That's what I'm showing. That whole thing is one lipid. Like all those sugars make up the head group of one lipid. So that one should be the easiest one to figure out what it is. If I like either asked you, what's a ganglioside and showed you a bunch of pictures, or if I showed you a bunch of pictures and I said, pick out the ganglioside, or if I had an essay that says, what is different about a ganglioside or what's special about a ganglioside, the head group has like 5 sugars on it. So that one's probably in my opinion, is the easiest to point out. Could I draw a cerebroside? Sure, should be able to. Let me clear all this. So just imagine that this, whoops. Imagine all this wasn't here. And instead, we would have like, how would that be? So it'd probably be like CH. No, okay, there it is. I found, let me do this 1 sec. Put me on the spot with my drawing. Yeah, so yeah, so it'd be like connected to the corner. There might be another oxygen there. And then we have our, okay, sorry. Trying to do this from memory is not the greatest. Oxygen, carbon. There we go. Then your sugar ring would be proper. Where you have like OH, OH, OH. That's basically a cerebroside. You just have one sugar there. I suppose it would have been easier now that I look at it, if I would have just said, yeah, it's basically that without the salic acid. That's probably I should have just done that. That's a cerebroside basically. Yes. So this green part, that's salic acid. Remember, these things have like 3 head groups, more or less. It depends. So the ganglioside, I would say, have 2 head groups. It has all of these sugars, which I would call a head group. It also has salic acid, which I would call a head group. And then you have the tail, the lipid tail there in the black. Right? I would define a head group that's not anything that's not a lipid tail. That's how I would define it. Yes, how would you describe a ganglioside? It has salic acid and it has like 5 sugars on it. That's how I would describe it. That would be my description. And that's all I would be looking for, to be honest. Like if the question was, describe what is a ganglioside? Okay. It has a head group that's salic acid. It has a head group that's 5 sugars. It's part of the sphingomyelin family. There you go. It's a sphingomyelin with salic acid and a bunch of sugar head groups. That's all I would do. All right. So speaking of sphingolipids, let's see how much of that you retained. And of course, I have a poll. Here's a question you might get on a test coming near you. Whoops, I messed up this poll because there should be more than one answer. All right, so this poll is weird. It's going to be wrong. But what I want you to do is treat these like A, B, C, and D. I forgot to write A, B, and C, and D, but like answer 1 is A. Answer 2 is B. Answer 3 is C. Answer 4 is D. Ignore the actual words. Just imagine it goes A, B, and C, and D because there's multiple true statements. So what is a sphingolipid? What statements best describe a sphingolipid? And the good thing about questions like these is it also reminds me of stuff I forgot to tell you, because I forgot to tell you something about these sphingolipids. All right. So I will call it there. We basically just have an even spread of answers. So let's just go back and briefly look at this. So A, does a sphingolipid contain a glycerol core with a phosphorylcholine head group? Yeah, going back. Here is glycerol all the way in the back. CH2, 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 OH, 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 right? So that is glycerol. Choline is CH2, CH2, and CH3. So we do not have that. Not only do we not have glycerol, we don't have choline either on our sphingolipids. This is the head group of a sphingolipid. That does not have glycerol. That is not glycerol, and it doesn't have that. So A is untrue, or our first true and false is not true. So that statement is false. That takes away A. Contains a modified sphingosine. So let's go back to our slides there. A sphingosine is right here. And yes, sphingolipids have sphingosines. That's what makes them a sphingolipid. So that is a true statement. Isn't sphingomyelin a sphingolipid? Yes, it is part of that class. They don't have glycerol though. So contains a modified sphingosine. Yeah, that's true. The thing I didn't tell you, I didn't emphasize or say at all during these slides, is that these things are generally receptors. That is their general purpose in the membrane, is that they are cell surface receptors. So they will interact with like proteins. That's generally what they're going to do, and so it is that. So this question was not very fair in that I didn't tell you all the information, but hopefully you could see that, well, 2 isn't every one of those answers, right? But hopefully you can see, yes, it contains that receptor. I just told you functions as a precursor. Not really. We don't take sphingolipids and like make them into anything. Later today, or early Friday, we're going to talk about precursors and show you exactly what a precursor is. But a precursor is like a building block. So we don't take like amino acids are more or less precursors for a protein, kind of. Not really, but kind of. For a precursor, is basically you're taking multiple sphingolipids and you are either modifying them a lot to create something new or like sticking them together. That's not happening here. You just have a head group and you're maybe changing the head group, but you're not like changing the whole lipid. So that's why I would go 2 and 3 here. Yes, sphingomyelin is a type of sphingolipid. It can get a little confusing with like all the names. So let me just clear that up, as that has been confusion in previous years too. A sphingolipid is like the general umbrella, right? This is the head group of the sphingolipid. Depending on how you change these, you can get different types of molecules, right? So if you add the fatty acid tail to the second position and you don't modify anything else, you have the sphingo sign, okay? Going forward from that, if I take my sphingolipid and add a phosphatidylcholine head group and I make a specific fatty acid, that is a sphingomyelin, right? If I instead have one sugar and a fatty acid here, that is a cerebroside. If instead I have a salic acid at my second position and this large, big old sugar head group as well, that is a ganglioside, right? So that is the definition. Like a ganglioside is a sphingolipid. Not all sphingolipids are gangliosides. So hopefully that clears up some things. Can you make a slide that has all those differentiated? That is, so that's what these two slides are doing, right? Top left, you have a sphingolipid. You modify the second one, that's a sphingosine. You take your sphingolipid and instead you modify it like on the left, that's the sphingomyelin. I don't have a cerebroside posted, right? But you just put a sugar head group, that's the cerebroside. You modify your sphingolipid here and you, to the bottom right, you make a ganglioside. So I won't make that slide, but I think it's a good study tip if you can try and make that slide based on the information I have presented, right? Because if you can make that slide instead of me making that slide and you memorizing it, the art of the process of creating that new flowchart of information will solidify it way better than me making it. If you want to make it and see and correct and see with me if it's right, I'm happy to do that. But that's my thought process as to why I won't make it, but yeah, you can definitely make it. I can check to see if the information you have there is correct. That's the compromise I'll make for that idea. Okay. So I have other questions here. So I will since we've gone long on this, I will post the answers up on Thursday. That's when I usually put up all the answers. And so go back to these before you look at the answer key and look at the information in the slides and see if you can answer 7 and 8. If you don't get it correct, we can talk about, send me like a message as in like, I got this wrong and I don't know why. And we can have that discussion moving on. But yeah, I won't reveal the answers for these, in that I want you to think about them a little bit. So that's going to be one way to test yourself again. Some more multiple choice, see if you get it right. If not, come back to me and it's like, why did I don't understand this concept? And I can definitely have a dialogue about that. But I do want to move on. I don't want to get too far behind again. since we've already spent some time on this, and talk about our work today. And what we're talking about are, first off, steroids. And steroids are usually only found in eukaryotes for the most part. And they're made out of, let me see if I can pronounce this, cyclopentanoperhydrophenethrine. Yeah. It means that. So that long word is the organic chemistry word for 4 fuse rings. I'm not going to ask you to know that word because that word is ridiculous. Our most abundant steroid in our body is cholesterol. I'm sure you've heard about cholesterol a lot. And those of you who take Biochem 2, I will tell you all about why cholesterol is bad for you and how to get rid of bad cholesterol and how to get rid of those blockages in your heart that you might have growing right now. But that's a discussion for next semester, those of you who are interested. But cholesterol make up about 30 to 40% of the lipids inside your plasma membrane. And these things are very important. What cholesterol does is that it will help change how fluid your membrane is, right? Remember on Monday, or it might have been Friday, I forget exactly when, we were talking about how membranes want to be fluid / solid like, almost like a milkshake. And eukaryotes can do that by either increasing or decreasing the amount of cholesterol in their membranes. Because if we have a plasma membrane lipid, or a lipid membrane, right? So here's my lipid membrane. The more cholesterol we have, and the way to think about cholesterol in the membrane is like, the way I think about it is like paddles or planks of wood. And so you insert these planks of wood and they push your lipids together. So the more cholesterol you have, the more you're pushing your lipids together, the more solid-like it gets. When you remove cholesterol, let's say if I just erase these, then, oops, I erased my head groups. Then your lipids have more space to spread out. They're not being pushed together anymore. So lower the cholesterol, the more liquid your plasma membrane becomes. They don't have as much things forcing them together. For us, we form cholesterol esters. So we give them a little baby polar head group, and that's to allow them to be in the plasma membrane. Plants, they don't really have cholesterol. That's more of an animal thing. They have other steroids though. So how are steroids and lipids in the plasma membrane similar? How are they different? I will answer this. So it's the way that steroids, and just because I say steroids does not mean they're bad. I'm not talking about the steroids that will give you like super muscles. Well, actually, steroids don't give you muscles. They allow you to train. That's why people get big on steroids. But anyways, I'm getting off topic. Steroids and lipids, they're similar in that they have a, let me get my pen back out, like a polar head group. I mean, cholesterol is a baby polar head group, but it's technically still there. And non-polar body. That's how they're similar. They're different by their body, right? Lipids have this long carbon tail. Steroids steroids are always going to have these four rings. That's what makes a steroid more or less, is that they are derived. And when you make the steroid, you have the six-membered, six-membered, and five-member ringed all fused together. That's what a steroid is. So similar in that polar high groups, similar in that non-polar bodies, different in what those bodies actually are. So cholesterol, we can use that to make other hormones. You have your glucocorticoids or cortisol, and these things will control a lot of stuff. So they'll affect your carbohydrate production, protein production, lipid metabolism, your inflammatory reactions, stress. I'm sure right before my test, I can tell if I go into any of the Proctor U videos, I can tell the cortisol levels in all of y'all are super high by the way you look and the way you're acting and all that good stuff. So when you get stressed out, you're just flooding your body with these steroids, but they control other things as well. You have your aldosterone, your mineral allocorticoids. They control salt and water in the kidneys. Your estrogens, sexual development. These are made in your adrenal gland. And what steroids really are, it's the way your body is able to communicate to itself on a large scale, right? So let's take cortisol, for example. Let's say you are very stressed out. You are going to produce this signal that is going out to the rest of your body and saying, hey, we're stressed right now. Maybe, start producing some more energy, start doing some more metabolism, because if we're stressed, we might need energy very soon. So that might be an example. Or, for your estrogen, oh, our body, yay, we're 12, 13 now. We're ready for sexual development. Start flooding your body with estrogen to tell the rest of your body, hey, we're ready to go now. Let's get real. So that's what steroids basically are. They're messengers for everybody. Why do you gain weight? I think that's with, so I'm not 100% sure, but I think it has to do with water retention is one part. And also some people have the coping mechanism of stress eating. And I might be wrong on the metabolism. You might just say, when you're stressed, store energy. I might have that confused off the top of my head. But yeah, I actually don't have a great answer off the top of my head other than I think you retain water because you're releasing those steroids, and a coping mechanism is to get food inside you. Maybe again, like saying, hey, we need energy. Let's start getting that energy. Oh, I'm hungry. Pump, 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 pump. We also have some vitamins. Vitamins, some of them that we make are created from steroids as well. So vitamin D is made by us, and they're from cholesterol. They're steroids. D2, this is the reason why you need to go outside every once in a while to get that UV light. You don't need much though, like 10 minutes, 3 times a week is all you need to get the vitamin D2 in your body. D3 is made from 7-dehydrocholesterol. Don't need to really know that piece of information. Just know that vitamin D made from steroids. D2 and D3 aren't active until you have an enzyme to turn it on. And one of the things that vitamin D does is that it increases the amount of calcium that you uptake while eating dairy. So that's why you always see on milk with vitamin D. They're trying to say, hey, we have this extra vitamin, so when you drink our product, you can get more calcium. Even though there's other ways to get calcium, because milk is gross and nobody will tell me anything different. Milk is the grossest substance on earth. I don't know why people drink. Ugh, ugh. You don't want to go outside. Does light that goes through a window work? Some windows do, some windows don't, right? So if your window has UV, has coating to protect these UV lights to go through, it's not going to do anything. Why some people say that milk's actually bad for you? I haven't heard that milk is bad for you unless you drink a ton of it. It's also not necessarily good for you. I mean, we can get by just perfectly fine through a diet of other things than milk. What are some alternatives? Cheese is a good alternative. Dark leafy vegetables like kale have a good amount of calcium in them. So that's an alternative, right? Yeah, those, I can't think of other dark leafy vegetables off the top of my head. Spinach, I believe spinach has some good amount of calcium, if I remember right. Again, I'm not a dietitian. I'm just trying to remember what I read here. Milk has a lot of sugar. Yeah, it does have a lot of lactose. I mean, and if you drink enough of it, your body will start to say, please stop doing this. Next time you drink milk, I'm gonna make you throw up and have diarrhea. So you become lactose intolerant. Funny thing is that, Europeans, people from European heritage, are really the people who benefit the most from milk, in that they become lactose intolerant the least, while you have like Asian and African populations that they can become lactose intolerant super easily. So that just goes to show you that don't believe marketing campaigns that say, oh, you need to drink milk or your bones are going to break down. Eat a balanced diet of fruits and vegetables. And there's my dietitian speech for you. All right, here are the precursors I was talking about though. So isoprenes, various vitamins that are built from isoprenes are made from this unit, this 5-carbon unit. So one type of enzyme that you might have heard about in your other classes, coenzyme Q, which is part of ATP synthesis. It's one of our electron transport molecules, coenzyme Q or CoQ, that is made from isoprene. So isoprenes, they are components of the membrane. They're not structural though, so they're not like a lipid, but they do other things, like I said, can carry electrons in oxidative phosphorylation. Ubiquinone, yep, that is another word for coenzyme Q. You can also buy coenzyme Q at the vitamin section. I have no idea what it would do for you, because you need coenzyme Q inside your body, inside your cells, inside the mitochondria, but you can definitely buy it. I don't know if it does anything. And here are some other ones. Retinol. So retinol, or vitamin A, is the thing that makes you see. It's a very, one of the things that make you see, I should say. It's a very cool molecule, right? So the way that retinol works is that it has like all these double bonds. When you see light, what happens is that light molecule interacts with these double bonds and basically changes the tail of vitamin A. That change in that tail, your body recognizes that as light, and we see stuff. That's very, very, very basic way in how you see. The light hits a molecule in your eye, goes through a reaction because of that energy. Our body recognizes the reaction and says, oh, that must be light. We have vitamin E on the bottom left here. And so it's an antioxidant. What an antioxidant does is that it will absorb electrons that prevent damage to membranes and lipids. And you'll see food labeled with this all the time. Contains antioxidants. I was reading a study, oh, I want to say during the summer, I was reading the study. And the study basically said there is no evidence that we can find that eating an antioxidant will actually improve your body's ability to fight antioxidants. So again, this is one of those warnings I like to give. Just because we know of something, like we know antioxidants work inside the cell, doesn't mean we can just eat antioxidants and they're going to do that thing, right? So just keep that in mind. We also have vitamin K over here on the right. Vitamin K is one of these things that help in clotting our blood. So I just have one more question for everybody. It's not on an actual slide and it's not going to be on the test, but just see everybody's general knowledge. So vitamin A helps us see. What vegetable are we told that we should eat if we want better vision? Carrots. Yeah, so fun fact, that's kind of a lie. It's true that carrots have vitamin A and beta carotene. Both of these are known to be in the eye. But eating carrots won't make you see better, and eating carrots won't restore eyesight. It's important that you eat carrots to replenish it so it doesn't get any worse. That comes from World War II. So in World War II, and this is just a fun fact, and I'll let everybody go. In World War II, the British had radar, before radar was a thing. And so they were shooting German planes out of the sky at night. Like the British fighter pilots were able to find the German planes and shoot them down. Oh no, my internet, please don't go away. And yeah, it was a secret message. And so instead of saying we have radar, the British said, well, we just eat carrots. And so we can see better at night. And what do you know? 70 years later, 80 years later, people still think carrots will make you see better at night. Yeah. A little misinformation goes a long way. All right. Well, that's it for today. I will put up a homework. And yeah, I will see everybody hopefully on Friday. Take care, everybody.